Sorry, still waiting for the little spinny thing to quit. There we go. Okay. Second half of what I've got to say about Platyzoa, and then we'll be done. So going back over to the slides. There we are. So we've talked about Rotifera plus Acanthocephala making up this larger group called Cindermata, which is what you see up here. Not to be confused with Cinderella now. So we've covered Platyhelminthians. The next group is not one you're likely to spot. They're all marine, and they live in rather unusual habitats. They're called gnathostomulids. Gnatho meaning jaw and stoma meaning mouth. Gnathostomulids means little things with jaws in their mouths. They are superficially like flatworms. The difference is that you have only one cilium per cell on the epithelium. True flatworms can have multiple cilia per cell. And they've got complicated jaws. And gnathostomulids were only discovered in 1956 because nobody had thought to look there before. They are among the most anoxic tolerant of animals, although we now know they're not the most anoxia tolerant. You'll meet those later. But they can live in habitats with very low oxygen that are rich in sulfide, and they live among sediment grains. They can creep through the spaces between adjacent sand grains, and the organisms that can do this have a name. They're called meiofauna, and you'll see that word again. Meiofauna is the set of animal species that lives in sediment in and among the very grains of sand. They're all very small. And so you'll find these in pretty shallow waters, typically on beaches, but not the kinds of beaches that have, bright yellow or white sand and lots of waves churning the sand up, because those tend not to be very anoxic. They tend to live in quiet waters where the sand tends to turn dark and smell funny if you dig into it because it's rich in sulfide. So beaches with a lot of organic decay consuming the oxygen and producing this, smelly, dark, sulfide-rich habitat, that's hog heaven for gnathostomulids. And although they look more like flatworms, the latest mitochondrial DNA data that I was able to find puts them closest to the Cindermata group we just talked about. There's one kind of a generalized diagram on the left and below. To the right, there's a specific one, Probognathia, with sensory bristles up at the front, a mouth, and then just back of the mouth, you have a whole bunch of little teeth surrounding the pharynx, and they're kind of folded up. They can be extended out of the mouth to catch food particles and then retracted to chew them. Back of that, most of the body's taken up by a gut. I don't think there's an anus here. The way out may be the way in. And then back of the gut, you have eggs and sperm. I have never seen one of these alive, and I'm pretty sure not many people have. But the reproductive system looks vaguely like that of a flatworm, where you have, eggs produced in an ovary. The testes connect to a stylet, a little piercing needle-like structure that I presume is used for sperm transfer. So there. There's another one. You can very faintly see those sensory bristles up at the head end, up at the upper right. back of that just a little bit, I can see where the jaws are located. And again, you really have to go looking for these guys to spot them. The second one's even less well known, and they were only described in 1995. I was in grad school when these were found, and if there's a lesson to be taken from this, it's that we haven't worked it all out just yet. There have actually been a couple of cases of proposed phyla being discovered after 1995, and it's simply a matter of finding a place where nobody's ever thought to look and, well, being willing to look. And we don't know much about these because there are only 3 species known. They're all in one genus, genus Symbion, and the only one that's been studied very well lives only on the mouth parts of Norway lobster. The other species live on other species of lobster mouth parts. So yeah, these are specialized to live only on lobster lips. Somebody's got to. You know, if you have worms that live in hippopotamus eye sockets, you have to have something living on lobster lips because the world wouldn't make any sense otherwise, would it? Right. They are sessile, glued to the bristles of lobster lips. The gut is U-shaped. There is a ring of cilia around a mouth that's shaped kind of like an upside-down bell. And this is maybe like rotifers, but there's no corona, there's no mastax. It's not really that much like rotifers. The life cycle, on the other hand, is crazy complex. It alternates between sexual and asexual stages, but there's something like 40 different stages in the life cycle. And the alternation seems to be timed to the lobster's molt cycle. Lobsters, like all arthropods, shed their cuticles periodically, much like the cicadas do that you're going to be seeing screaming from every tree in probably about a month. Screaming chicken nuggets, as our cats call them, because they try to eat them. Anyway, and when lobsters shed their cuticles, that includes the cuticle surrounding the mouth parts, which means that if you're living on a lobster, you get discarded on a regular basis whenever that cuticle is shed. So cycliophorans create ciliated larvae that can briefly leave the mouth parts and then settle back on them when the timing is right. And that seems to be why the life cycle is kind of strange and complicated. The males are dwarf. They do go through a sexual cycle. And as we saw in monogont rotifers, the males are dwarves. And that's what one of these looks like. That's a female cycliophoran. Down at the lower right, that's the sensory bristle of a lobster mouth part. And so she is kind of attached to this thing by a sort of stalk. The body is kind of sac-shaped. And up at the upper left, you have this funnel-shaped or bell-shaped mouth with cilia surrounding it, leading into this U-shaped gut with the anus looping back and coming up, opening just behind the bell. Attached to her, we've got a dwarf male, and he's 40 microns long. That's one of the smallest animals known. And at sexual maturity, he consists of about 50 cells. So evidently, this whole multicellularity thing was just too much. You know, he's stripped down to producing sperm, and that's all he's good for. Here's what they look like on an actual lobster lip bristle. These are scanning electron micrographs. Down the one at the left, you can clearly see that funnel around the mouth with the cilia surrounding it, the thing shaped like a bell. I can see the anus looks like a little slit just behind that bell, the kind of lump-shaped body, and then there's a dwarf male on, from our point of view, the lower side between the animal and the lobster bristle. And that's how abundant they can be. That's some piece of a lobster mouth part that's covered with Symbion paradoxa. And the life cycle gets bizarre. And don't worry about the details. You'll have an asexual stage that produces larvae that can swim and then settle on the same host. So one that colonizes a lobster lip can produce many offspring asexually on the same host. But then periodically they will start producing males, reproduce sexually, and give rise to a special type of larva called a chordoid that can settle on a new host. And again, that's probably adapted to escape the lobster before it molts and then colonize that lobster or a new lobster afterwards. So again, don't worry about all the details, though. Just know that it is weird and complicated. Okay, right. Gastroctrix you might actually see. One of these days, I'm thinking about buying a live lobster at Bougie Kroger over on Salem and ripping off its mouth parts and seeing what's there. But I just have never really felt like it yet. But Gastroctrix you could spot around here. They're pretty common in the local freshwater. The evidence from DNA seems to place them near other Platyzoan taxa, but nobody seems quite sure. There's at least several hundred species of these. They're all microscopic. They're all aquatic. And you tend to find them in the same kind of habitats where you'd find rotifers, although they're not rotifers. There's a mouth, there's an anus, a simple muscular pharynx pumps and brings tiny food particles in through the mouth. These move by cilia that cover the belly, the ventral side. Gastrotricha is Greek for hairy stomach because they've got these cilia. And then the entire body is covered with a protein cuticle that covers the whole body and also covers the cilia. And this often might produce little spines or things like that. There's virtually no coelom. The body is just filled with organs. And many freshwater gastroctrix are parthenogenetic and a syncytial epidermis. That's hauntingly like rotifers, but whether it's convergence or not, I suspect it's probably convergent evolution. And there's a fairly typical freshwater gastroctrix, Chitinotus. You're looking at the external anatomy on the left. What you can see is that that protein cuticle makes up some scales and spines and things like that. The whole thing looks a little spiky. On the right, the mouth is at the very tip. You've got a fairly good-sized brain in the head. The pharynx is that kind of stripy-looking thing back of the mouth, and it leads to a stomach that I think is made up of roughly 20-odd cells. Connected to the stomach, you've got a couple of excretory organs, those tangle things. Back of that, you've got ovaries, and in that forked tail, there's a couple of glands that secrete cement, an organic substance that allows the gastroctrix to kind of glue itself down. That's what they look like. These are both taken with a type of microscopy called dark field, which is why it looks like they're glowing. They superficially look like rotifers, but they don't have a mastax, they don't have teeth, they're not segmented, and they don't have oral cilia on a corona. So yeah, they don't have those cilia on the lobed organ that appear to be rotating like a wheel. They haven't got that. You can see the mouth fairly clearly. It's just that little, oval-shaped thing right at the top end, and you can see those forked toes. And the one on the right, you can see the spines. And then this happens to be a marine species. That one's got kind of a funnel-shaped mouth. Some of the marine forms have hooks in the mouth. On the underside, you can see the cilia that enable it to move, surrounded by those spines made of the protein cuticle. And on the back, it's got scales that are also made of that protein cuticle. Okay, we might see those tomorrow. Keep your fingers crossed. Chaetognatha are another group that nobody knows where the bleep they go. Exactly where is uncertain. The latest molecular evidence from about 4 years ago seems to put them near the gnathifera, but this could be wrong. They're all marine, they're all small, they're all predators, and they're pretty important in planktonic ecosystems because they'll feed on tiny crustaceans and then themselves get food for small fish and things like that. So they're economically important in the plankton in the ocean. About 80% of all species are planktonic, and they're very long, and they don't really swim in a sustained fashion, but they can flex their bodies and then spring forward, looking a little bit like an arrow that's just been fired. They've also got fins that look a little like the feathers on an arrow. So the common name for chaetognaths is arrow worms. I think I said they're all marine. We're not going to see any tomorrow. The mouth is surrounded by a bunch of hooked spines that can be retracted or extended to trap prey. Swimming is stabilized by fins on body, and the body's divided into 3 parts: a head, a trunk, and a tail. So what do these look like? They look like this. That's a very typical planktonic chaetognath. You can see those transparent fins that look a little like the fletching on an arrow or maybe the strakes on a missile. The gut is clearly visible inside the nearly transparent body. Up there at the head, I can see where the spines are, but you'll see that in just a minute. Chaetognatha, by the way, means bristle mouth. So these are the critters with bristles around the mouth. And then there's an SEM of a chaetognath head. When the thing's swimming, the spines are retracted under a hood. And there the spines are partly extended out of the hood. So the chaetognath can retract them for more streamlined swimming, or can expand them into this kind of spiky fan. Frankly, kind of like my cats do to me sometimes with their claws, that they'll use to trap prey and drag them to the mouth. So that's all the chaetognath stuff we've got time for. When I was in high school, we considered platyhelminthes to be at the base of the tree of life of the Bilateria, the most primitive of all animals that weren't sponges or jellyfish or something like that. There's lots of evidence from DNA and structure that pretty well confirms that platyhelminthes are not at the base of the tree of life. They're nested within the protostomia. The problem is there's some oddballs that are sometimes treated as platyhelminthes. They used to be, but they're probably completely separate phyla that don't share true common ancestry with platyhelminthes. And one of these is a group called the acelomorphs. They look like flatworms. They're small. They're mostly marine, although there might be some you can pick up in fresh waters. The extracellular matrix has been reduced. The brain is more or less radial but turned on its side. Most are meiofaunal, and there's that word again, that they inhabit spaces in between sediment grains. And there's a few that are planktonic. And one subgroup of them, the acela, has no gut lumen, no open space in the gut. The gut's a syncytium. The gut's a multinucleate mass of cells that just engulfs food particles and digests them there. As far back as 1999, data from DNA was placing these at the very base of the bilaterian tree, not with the rest of the platyhelminthes, which seem to be quite separate, up farther down the tree in the protostomia. Whoops, sorry. There's been controversy over this, so I could be wrong here, but last I looked, the bulk of the preponderance of the evidence was placing the acoelomorphs outside the platyhelminths and very close to the root of the bilaterian tree. So they look like this. Polyacanthus on the left and flagellophora on the right. If you look at the one on the right and you look about 2/3 of the way from the bottom, you can see a pair of what look like little balls on the head end. Those things that look like small, like the things carrying BBs, those are called the statocysts, and those are balance organs. And we've actually seen organs like that in jellyfish and ctenophores as well. This is the thing that looks kind of like I compared it to a golf ball inside a beach ball, right? You rotate the beach ball and the golf ball will always roll to the bottom. Well, the one on the right, the nemertodermatid, has two. The one on the left, just ahead of all that black stuff, which I think is the syncytial gut, it's not quite as obvious, but I can see one up in the head end, just ahead of the black stuff there. One very cool one that was a favorite of Lynn Margulis is Simsagitifera roscofensis, which is noted for not feeding at all because it's completely dependent on endosymbiotic green algae. And it's surprisingly common in the sand of Atlantic and Mediterranean beaches. It can be very common on some of the beaches of places like the French Riviera. So just saying, if you ever want to go, if you're looking for something to study as a grad student, it'll help if you can find something to study that only lives on awesome beaches. So yeah, just a tip there. Maybe you could get a grant to go study the French Riviera. Right, anyway. If you take a cross-section through Simsagitifera, this one doesn't have green algae. It's got probably dinoflagellates there, but that's a slice through a worm a lot like the one that you just saw, and you can see it's completely packed with endosymbionts, and there's no body cavity at all, including a gut. The thing's a solid mass of cells getting all of its nutrition from symbionts. And this is a molecular cladogram. This is about 18 years old, and I need to see if it's been updated. But if you can see those colorful boxes, the red box contains all of the protostomes. And within the red box, that's a green box, those are platyhelminthes. They're labeled Rhabditophora, that's flatworms. Platyhelminthes are all in the green box, and you can see their closest relative is that branch labeled Rotifera. The deuterostomes are all down in the blue box, and then that purple box is where acoelomorphs go. Those acial flatworms that we used to think belonged with the rest of the flatworms actually belong as the earliest branch of the bilateria. So not everything that we used to call a flatworm was in fact a flatworm. Very simple wormy beast here. A live one's on the right, but there's not a whole lot of detail to see, called Xenoturbella bachii. Used to be grouped with the acoels when anybody bothered to group it with anything. But some DNA studies have actually put it down at the base of the deuterostomes. So this may be the earliest branch of the deuterostomia, even though it looks nothing like a echinoderm or chordate. And what seems to be plausible is that very simple wormy beasts like Xenoturbella and, Synsagitifera and all of these things still retain something of what the very earliest bilaterian animals were like. That basically what you would see if you look down at the deepest base of the bilaterian tree would be some very simple acelomorph-like worms, and then their descendants specialized into the more familiar protostome and deuterostome taxa that we see. So modern acoels are not our direct ancestors, but they may turn out to give us some idea of what our direct ancestors looked like way down when the very first bilateria were starting to appear, call it very roughly about 600, 700 million years ago next Thursday. So there, profitable things to be learned even from the most obscure wormy beasts. All right, that's actually a pretty good place to stop. We've got a lab tomorrow, and this is one where I will work scope and camera and then turn more, and then we will finally be caught up on the labs. You've now got lab 6, so and you'll get lab 7 tomorrow, and we should finally be caught up, and with any luck, I will actually have some grades to hand back. And there's me. Do I have any questions before I turn you loose? I don't see anything on the chat. So I'll go ahead and stop recording now, and I'll see you folks tomorrow.